Okay, I think that the number of people that have been joining is slowed down, so we'll go ahead and get started. My name is Tyler Beck. I'm the Wetland Avian Habitat Specialist for Florida Fish and Wildlife Conservation Commission and chair of the Snail Kite Coordinating Committee. I want to thank everybody for attending. I know today is election day. I hope this scheduling of this meeting didn't interfere with anybody's plans to vote. But it was the best time for most of the people to be able to participate today. I want to let everybody know this meeting is being recorded, and that I will try harder this time to make that meeting recording available after this. And so if anybody wants a copy of the recording or access to it, I'll try to get that to you. But Teams does not make that easy to share outside of your organization. So, also I want to say, I'm sorry for those of you that were looking for the agenda when I said that the agenda was on the Teams meeting. It's there. It shows up on our end. I don't know why a lot of people were having trouble accessing it. But I didn't want to re-email it out to everybody for a couple of reasons. One is because every time I changed something in Teams, it would send a new notification to people, and then I would get a bunch of notifications that people were accepting the meeting again, even though they'd already accepted. So I didn't want to do that even more. And there are a lot of participants here that have accepted the meeting that are not on my email distribution list, so they wouldn't have gotten it either. So I just chose to send it to people that asked about it, and even regardless, we had a cancellation in our schedule yesterday, so the agenda isn't even accurate anymore. So, but I will share that with you in a second. So I'll talk a little bit about what is the Snail Kite Coordinating Committee and where it came from. I'm actually not all that super versed in the history of it, and I don't know if Dr. Gray is here to straighten me out on some of this stuff, but it was started as a conversation about, we really need a forum to trade information about what's going on with snail kite research, snail kite management, general snail kite, what's going on in the world of snail kites. And so it's kind of just a loose committee of anybody who wants to attend these meetings, and we exchange information about snail kites. We have a charter. I should have prepared that to share. We usually like to share it every once in a while so that people understand what the point of this meeting is. And I can send that to people if they want to see what the charter for this meeting is. So I mentioned before that there are a lot of people that have accepted this meeting that I don't have on my email distribution list. And so I would like people to know about this meeting that need to. So if you're don't get the emails directly from me about doodle polls for scheduling this meeting, or don't get the invites directly, and you want to get those, send me an email. My email address is tyler.beck@myfwc.com. I'll put that in the chat. And just let me know, your name and your affiliation, and I'll add your email address to my distribution list. And anybody else that you think that might be interested in attending these meetings, it's just an open exchange of information. Anybody can attend. There's no restrictions or anything. So yeah, share it with anybody you think might be interested. I want to introduce a couple of people. We have some new faces in the snail kite world, some old faces in new positions. So first I'll start off, she isn't super new. She was at the last meeting, but I'll let Monica introduce herself.
Thank you, Tyler. Hello, everyone. My name is Monica McKenzie. I am the bald eagle and snail kite biologist with FWC. I've been in this position since March, and I'm located in Kissimmee. Thanks.
Thanks, Monica. One old face, but in a new position is Brian Jeffrey. So I'll let him introduce himself.
Good morning, everybody. Yes, my name is Brian Jeffrey. I'm the Snail Kite Recovery Lead for the U.S. Fish and Wildlife Service. And before, I was the Snail Kite Project Manager for the University of Florida. So I was in that role for about 10 years.
Okay. I'm not sure if Dr. Rob Fletcher is here with us right now, but he was heading the Snail Kite Monitoring Program at University of Florida for a long time. He's now at Cambridge. And then sitting into kind of taking over that monitoring program for him is Dr. Miguel Acevedo. Dr. Acevedo, I know that you probably had your introduction into your presentation, so we don't have to go to you now if you have that already set up for you.
But I can just say hi to everybody, and that I'm super happy to meet you all here. And hopefully this is the first of many.
Thank you. We're glad to have you. So if you've been attending these meetings for quite a while, you know that, well, you may notice that my title has changed. I'm no longer the Snail Kite Conservation Coordinator for FWC. I've shifted roles a little bit. Wetland Avian Habitat Specialist. I still do have some role in snail kites and include supervising Monica in her duties. So I'm still involved in the snail kite world. But with that, I do plan to kind of step away from leading this committee in the near future. That doesn't include our next meeting. Our next meeting will be, we do have an approved session at the Greater Everglades Ecosystem Restoration Conference this spring. So anybody that's going to be at that conference, please come see our session. So Monica has helped me put that session together, and she's helped me with this meeting. She's basically been kind of a co-chair for this meeting. So we will kind of be transitioning some of my duties for this committee to her, unless anybody has any objections. We'll hear any of that. The other thing that is a big change with this committee right now is we lost our other co-chair this last spring, Vicky Garcia with the U.S. Fish and Wildlife Service. Unfortunately, she passed away due to a long illness. And we really miss her presence on this committee and her presence in the snail kite world. She was always involved with everything that required, guidance from the Fish and Wildlife Service and Endangered Species Act and anything related to conservation of snail kites. So her presence is very much missed. We are happy though that Brian was able to step in to her role recently and is now the lead biologist for the snail kite for the service. So we have also asked Brian to step in as a co-chair on this committee. So after the GEAR conference, I expect that Monica and Brian will be the new co-chairs for this committee, unless anybody has any objections or would like to assume responsibilities for this committee as well. But we were going to have a long, drawn-out conversation about that, but the more I thought about it, the less I thought that it was really going to be much of a conversation. So I'm just more informing the committee that that's the plans for this committee going forward. With that, I will share the agenda that we have put together. So this is the agenda that was supposed to be attached to the meeting invite. Unfortunately, we found out yesterday Dr. Wild is not feeling well, and she has a seasonal flu, and she will not be able to present today. With that, so we're going to kind of take our time with our two speakers, with Dr. Acevedo and Dr. Ken Meyer. If those talks, can stretch up until our lunch break, then that's good. We'd have as much conversation as we want. If those conversations end a little early, then I've talked to Megan Beatty and Laura about doing their presentations before lunch. So there might be some shifts in this agenda. And, if we wrap everything up and conversations have kind of waned early, then we will end this meeting early if that's what's necessary. So, but Dr. Wild also did say that she was willing to talk with us or reschedule her presentation. So I was thinking we could do a separate meeting just to give her presentation. Dr. Wild is with the University of Georgia, and she is kind of the expert on this disease called vacuolar myelinopathy. We used to call it avian vacuolar myelinopathy because it was first really showed up in birds. And it's a disease that's caused by a cyanobacteria that produces a toxin that can essentially eat holes in parts of brains of vertebrates. And it's shown up in that cyanobacteria grows on hydrilla, and things that eat the hydrilla can get it, and then things that eat those things that eat the hydrilla can get it. So although we've never documented a case of snail kites getting VM, it's always something that's on our radar. It's been a big problem with bald eagles in more northern states, but we haven't really shown issues with it here in Florida. So if you're interested in that talk, I have a poll that I'm going to try to put up. So I will gauge. I can figure out how to put up the poll. Where is it? Oh, there it is.
Tyler, Matt Phillips has his hand up.
Go ahead, Matt.
Hey, yeah, I was just going to suggest we have her scheduled to speak to our ASCT, I think November 14th, or at our next ASCT. I believe it's November 14th. I can check that, but that might be an opportunity to get additional folks to listen to that. Yeah, it would be the 14th, and I'm assuming we're going to do that first thing. So the 14th at 10 a.m. as an option.
Okay, but those are primarily like an internal meeting for FWC.
Yeah.
So I'll present that as an invitation to any FWC staff that are tuning in. I don't think we can really invite external staff. So
Sure.
Yeah. So, but thanks for that, Matt.
Mm-hmm.
Yeah. So if you're internal FWC and you would like to attend that meeting on the 14th, just shoot me an email and we'll try to get you an invitation to attend that. Monica, can we see the poll? Can you see the poll?
Yes, I can. We
Okay.
have 6 yeses and 0 no's so far.
Okay. I guess I have to fill out the poll in order to see the results. Okay. So just to gauge interest in doing kind of an external presentation for that, for Dr. Wild, it might be like a lunch and learn or something. It'll probably be, an hour. I think she asked for about 45 minutes for her talk. So I think it might be a good format for like a lunch and learn or, a quick hour presentation to learn about vacuolar myelopathy. They found it in a lot more than just birds recently, or she's found it in reptiles, and I don't know, I can't remember if she's found it in any mammals, but a lot of fish. So it has potential to have far-reaching, effects, and particularly if you work in systems that have a lot of invasive submerged aquatic vegetation like hydrilla. That's where it seems to really be an issue. So, all right. So, moving on to that, we'll go ahead and start with Dr. Acevedo's talk. He's going to give an update on snail kites and introduce himself.
Great, and you hear me?
Yeah, you're a little quiet.
Awesome.
But maybe that might be my speakers. Monica, how can you hear him all right?
Yeah, I can hear you. Can you hear me? Okay.
I can hear him
Okay.
okay.
I'll turn
All
my
right,
speakers
so
on. Thanks.
I'm gonna share my screen here in a second. I need a second here. It's asking me for, we don't use Teams here too much. We use Zoom. So my computer is screaming about some sort of permission. So I'll need a second here. I'll be back.
Okay.
Tyler, while we're waiting, I sent you a copy of the original charter that you can share with everybody. And as Nick said, this committee was formed kind of in the early part of the 2000s when the kite population very famously crashed and everybody was really scared, and we just wanted to get everybody in the room and share information. So anyway, that charter is in your mailbox somewhere, and you can share it with everybody just to refresh their memories. So thank you.
Thanks, Paul. As soon as I get that, well, looks like Dr. Acevedo's about ready. Oh, I got it. Okay,
Give me 1 second here.
sure.
All right, can you hear me well?
Yes, we can.
Yes.
Awesome. All right. So first of all, thanks for inviting me. It's really awesome to be part of such a great group. I'm gonna, let's see if I can, yeah, there we go. So before starting, I'd like to share a few things about my background. I know some of you, I don't know all of you. And first, I like to answer like a common question that I get, and people ask me, "Are you the one replacing Rob Fletcher?" And the answer is no. Rob Fletcher is irreplaceable, but I'm the lead PI in the UF monitoring program, and Rob's still very involved. But I'm here also part of a team. Here at UF, I'm a quantitative wildlife population ecologist, and I have a teaching and a research appointment. And in my teaching appointment, I joke around that my job is to teach math to wildlife students that, would much rather be outside. So I teach a bunch of different quantitative classes. In my research, I'm interested in three main things. I'm interested in populations at disequilibrium with the environment, and that's, for example, those that are dealing with climate change or land cover change. And I have students working with birds in the Amazon, mammals in indigenous communities in Ecuador, and we have a lot of work also in Puerto Rico looking at secondary forest recovery. And now I'm also leading the Snail Kite project monitoring. I'm also interested in diseases. Kind of bummed out that we couldn't see Dr. Wild's presentation. Here in our lab, we have students working with malaria in lizards. And the third part of my research program that I enjoyed a lot is that I work a lot with other quantitative scientists, like, for example, computer scientists, statisticians, mathematicians, and engineers, to develop models for conservation and management. I read somewhere that projects success is 20% what you do and 80% of the people. And I've heard only the great things about this group of people working, at UF, but also in the different agencies. So I'm super, super happy to be part of this team. So for those of you that are joining and not super familiar with our monitoring program, here in UF has been monitoring snail kites since 1996 in a very standardized way. Our monitoring includes surveys. It's basically two components. There's a very strong field component that has surveys, nest monitoring, and capture, mark recapture. But there's another like strong component of models. So we take this data from the field and we use the best models available to estimate those population level outcomes that we're interested in making inferences on, like population density, population abundances, survival, nest survival, and all that stuff. So the mission of our Snail Kite monitoring program is to provide information and guidance to inform decision-making for research managers and decision makers
Dr.
through effective
Acevedo?
research. Yeah.
Sorry to interrupt, but I think we're not seeing the progression of your slides. We're on your research and teaching slide still?
Still?
Yes. All right, so your mission statement? Okay.
Okay, let me just double check that. So I'm going to do something different then. So let's do this then. And I'm going to do, how about now?
Yeah, okay, we see the mission statement.
Perfect. All right, so basically our mission statement is to provide the best information available, the most accurate intelligence so that state managers can make the best informed decisions. And of course, if there's anything that I'm going to present now that it's really good, it's not because of me. I've only been in this position for a couple of weeks, maybe a couple of months, but it's because we have a really, really awesome team of dedicated people, including faculty, technicians, project managers, students, and all that. So if there's anything cool that I'm going to show, it's because of this awesome and great people. So I like to start sort of with a zoom out view and then go a little bit on the details. So what I'm planning to do today is I'm going to talk about the new 2023 field season, but I would like to put it in the context of the whole time series, if that's okay with you. So this is a figure that shows how superpopulation size have changed through time. In the orange dots are the year-specific superpopulation size estimates with their confidence intervals, but in gray we have that 3-year running average, which we believe it's better. It's a more accurate way of understanding how these things change through time. And as many of you probably know, there's a couple of different stages in this time series, including the first 2 stages of decline up to 2009, 2010, where there's an increase. And now in the last couple of years, we've seen that superpopulation size to be somewhat stable. And our estimate for 2023, it was somewhat similar. The mean estimate is somewhat similar to 2022, around 2,800 birds. And note that this comes from a somewhat sophisticated model that includes a lot of nuances that are required to make sense of the raw data, including probabilities of detection, immigration, emigration, and all that. The other interesting sort of trend, zoom out trend here that I want to show is population growth rate. In this case, we estimated population growth rate as the superpopulation size in time t+1 divided by population size in time t. And if that division is equal to 1, then the population is stable. If it's greater than 1, it's increasing. If it's greater than 1, it's decreasing. And as we see through the time series, the snail kite superpopulation has been sort of going up and down with some times in the time series where it was below that threshold of 1, in which the population was decreasing, some times in which it was above, and now it's somewhere around that threshold condition of 1. The mean estimate it's slightly lower than 1, but note that 1 is still within the confidence intervals of that mean estimate, and also is within the confidence interval of the 3-year average. So going now a little bit deeper about what's causing those fluctuations in that superpopulation estimates. Here we're showing apparent survival through time. In the white dots we have adult apparent survival, and in the black dots, we have those juveniles that are greater than 30 days old. And there's two take-home messages here. In the adults, we see some variability, but in the last couple of years, there's a little bit more stability on their apparent survival. Juveniles have been somewhat decreasing lately. 2023 is showing a little bit of an increase, but compared to the other times, is still quite low. Another interesting thing about the way that our project models survival is that we incorporate the probability of detection for survival. And that's really important to show that those fluctuations are not necessarily because, our ability to detect or not the kites changes, because the model is actually actively incorporating that information. But still, we see here our probability of detection have somewhat increased through time, and in the last couple of years, and it has been pretty stable, which is good. In terms of nesting, it's kind of interesting. Maybe one of the take-home messages of what we're seeing with the snail kites is that there's a lot of temporal and spatial variability. And I really like this figure because it shows how the nesting, for example, varies a lot in time, both across years, but also within the season. We start here, this is sort of before 2002, sort of in the 90s, and this is a figure here more of the recent breeding seasons. And we note a couple of things. One, that the peak is somewhere in the similar places, but there's a lot of variability across the years, with some years actually having kind of like two peaks in the breeding season, where other seasons only have the one. So we have a lot of variability among years in terms of the duration of that breeding season, but there's also a lot of variability within a week, which is kind of interesting. In terms of fledglings, these are raw counts of fledglings, and we've seen that they're varying through time. There was an increase after 2013, and a lot of variability jumping up and down until our earliest estimate of 2023, which was lower than 2022, but not super different compared to the other ones in the previous years. But note that these are raw counts, so I wouldn't make a lot of inference based on this, because to really make appropriate inference on this should be inside a model with estimated confidence intervals and incorporating probabilities of detections and things like that. Now, here, we're looking at the number of fledglings by size. So now, instead of looking at a temporal variability, we're starting to look at more spatial variability. And the pattern might actually be known to many here in this meeting. Here in the Kissimmee River Valley, there was an increase through time with a lot of variability after 2010, and our estimate from 2023 was slightly higher. And again, these are raw estimates, slightly higher than 2022, but actually in a pretty similar ballpark. The Everglades are still decreasing, and our estimate for 2023 is pretty similar to what we observed in 2022. Now, there was some differences here in the STAs, and note that these are regions, right? So, and these regions include a couple of different lakes. Lots of variability here in the last couple of years, but the estimate of 2023 was lower than 2022. In St. John, also some variability increasing in the last couple of years, but the 2023 was somewhat lower than 2022. Okeechobee, it varies a lot through time, and Lake Okeechobee in the 2023 actually was higher than 2022, showing that in this particular case it was an interesting source of populations. And then the other ones, there's a lot of variability here after 2013. In terms of apparent nest success, we see again a lot of temporal variability. The last couple of years, it's somewhat stabilized, and our estimate for 2023 was pretty similar to that of 2022. Now, this is a table that Lara prepared for our nestings in the most recent breeding season of 2024. And we see a total of 251 active nests, and out of those, we have known phase for 236, with 96 being successful and 140 that failed. So we can see here some of the estimates for nest success. I wouldn't worry too much about this one, and this one, because these are estimated based on a very limited number of nests, so they're not super accurate. But if we look at the other ones, we see that there is a lot of spatial variability on apparent nest success, with some places having someone on the lower end of 0.13, 0.33, and other places that did a little bit better. Average nest productivity, and again, I wouldn't worry too much about this, because the number was not, the sample size was not large enough to have an accurate average, but the other ones are somewhat consistent. In terms of nesting and water depth, in general, we know that water depth is really important for nesting and nesting success and all that. But this is a really interesting figure from our field season in 2023 in Lake Okeechobee. In red here, we have the water levels and how they change in 2023. In gray, we have nesting, and we see a very interesting pattern here that shows how these two things are very related. In terms of nest productivity, it's somewhat stable. In terms of there's not a lot of spatial variability. There's uncertainty, particularly in these lakes in Paynes Prairie, Lake Toho, because there were not too many nests over there, so the estimates are a little bit more uncertain. But here in Lake Okeechobee and Perscado, those estimates are a little bit better, but they're pretty similar. Somewhere here on the lower end on Paynes Prairie, and that's something that we're gonna look at a little bit more closely now with the 2024 data. One of the interesting things that is emerging from our understanding of the long-term trends here in the snail kite monitoring is how this population level outcomes are changing with the invasion of the other snail. And we see here that we estimate that breeding probability actually it's decreasing after invasion, similar with the survivor probability. Nest success have a similar, somewhat like a lower slope on that trend, but still somewhat decreasing. And the migration probability is also shifting through time after the invasion, where it sort of maximizes here an intermediate, but then it sort of decreases and for adults, and then for the juveniles it sort of levels off, which is starting to suggest that the potential positive effects of the invasive snail may actually be decreasing or leveling off right about now. And the thing is that we've been seeing this in multiple ways that we look at the data, right? We see here multiple different outcomes, but also there's a PhD candidate in our lab that it's building a population viability analysis for Paynes Prairie. So his idea for this chapter of his dissertation is actually comparing how those persistence probabilities can be estimated from eBird data and how that compares with the standard monitoring. But he's actually showing a very similar pattern that what we're showing with the previous data on the snail kite monitoring. We have somewhat like a decrease in the probability of persistence after 2020 and 2021, and then somewhat leveling off after that, or maybe even decreasing. So there's a hint there that the potential positive effects of the invasive snail might actually be saturating or leveling off. Just a quick update on the movement patterns. So we tagged 30 fledglings, and out of those we found 15 dead, and 7 of those tags were recovered, and 4 were presumed that died because of predation. They travel a lot, not surprisingly, with an average of 158 kilometers, but there's a lot of variability, right, on that estimate. It was interesting, we found a bird that traveled pretty fast, 88 kilometers per hour, almost 55 miles per hour in some roads. It might actually get a speeding ticket. They have relatively home ranges, 0.08 square kilometers based on a 90% kernel. And birds remain relatively closer to the nesting sites around 1.7, within 1.7 kilometers for 48 days after fledging, but also know that number also has some variability there. So what is the future holding? So based on our 2023 data on using the number of nests of the previous years or the number of observed fledglings, we are predicting a hint of a decrease in the 2024 data. So a couple of take-home messages from what we've seen in 2023. There's a lot of spatial temporal variability, both in terms of nesting and also in terms of densities and survival. That apparent positive effect of the invasive snail may actually be sort of leveling off, and our data is starting to maybe hinting of a decreasing trend that, of course, needs to be confirmed by the models and now with the 2024 data. And with that, of course, I would like to thank all the people involved here. Many of you are here in the call, and also thanking the funding for this project. With that, I think we can have a discussion, answer questions, or whatever you have in schedule.
Go ahead, Beecham. Beecham, you're muted.
Sorry about that. Can you go back to the slide that shows Lake Okeechobee and water levels, please?
Yeah. Give me one second. This one?
Yes, yes, sir.
Mhm.
We talk about this a lot. We talk about how water depths affect the birds on Okeechobee. But I mean, my question is, it appears to be obvious. Oh, as water levels go down, nesting goes down. And that's okay, number of active nests. With the change in the water level schedule regime on Okeechobee, where it is possible that Lake Okeechobee could be kept at higher levels, albeit we would love for it to get down lower during the year. But if the levels of the lake aren't this dramatic, if it stays around 15, 16 feet going later into the spring, does that mean that you're going to have birds that will continue to nest into late April, May, even into June? And I mean, that, I know, we talk about this a lot. I know that this was discussed when we went through the LOSOM process, but it appears that if you stabilize water on Lake Okeechobee, that you could have a longer nesting, maybe even more successful nesting while you're killing the lake. I know that higher levels will kill the lake, kill the marsh eventually, but will more stabilized water levels, keeping the lake around 15 or 16 feet, going to help snail kite nesting. And that may be a naive question, but it's interesting to me.
Yeah, so I haven't been here in the project long enough to develop an intuition that allows me to predict what's going to happen when that happens. I know that sometimes when we have like a big rainfall events, we can have, and I show it in the previous figure, we can have like that second sort of peak in the breeding season. But I don't know if Brian and Lara here have some insights. They've been working on this longer, and maybe they have that intuition that allows them to predict what might happen if those levels are kept at that level a bit longer.
Sure, I might be able to give a little bit of insight here. So Rob did a threshold analysis in the early 2020s, and the lake levels don't necessarily need to be stuck at 15 feet. The initiation models shown that the snail kites will initiate nesting anywhere from 12 to 15.8 feet. So getting stuck on, I'm not saying you are, but a lot of people do get stuck on keeping the water levels at this 15-16 foot height so snail kites can initiate nesting. They will initiate nest at a lower level. The biggest thing is when you have these drastic recession rates, that the water underneath these nests will dry out, and then that'll lead to nest abandonment or predation from terrestrial predators. So we have seen a larger increase in nest mortality. So if they want to kind of start the lake at 15 feet in the beginning of the year and gradually bring it down, I mean, that'd be great. But there are constraints as far as flood control and other metrics. But the biggest thing is, once the snail kites are nesting, is not dropping those water levels very, very sharply to where these nests are failing. I hope that clarifies. I might have gone on a
No,
little
and
bit
that's
of a rant.
exactly my concern, is that somebody looks at that figure and says, oh, well, let's just not drop the water levels in the lake. We'll just keep them high. We'll keep them level through the spring so the birds can be successful. And I saw Paul jump on as soon as I asked that question. That every scientist that has gone through the Lowson process says you're going to kill the marsh if you keep the lake at 15 feet. But if you're looking at it from that perspective, oh, well, I'll just keep the lake at 14, 15 feet. I'm not going to drop it. I'm just going to let the birds continue to live. And that is looking at a single species, obviously. But that,
Right.
I mean, that's part of my concern when people look at that.
Yeah, the chart that Miguel shows, it's only 1 year, and the kites are going to respond to how the water levels are. So, for instance, like this year, the water levels were really high. I mean, we saw nesting in southern Moonshine Bay, which typically doesn't have nesting just due to shallower waters. And, it was an extreme event because we had some tropical systems from 2023 contribute to higher water levels. But, the kites are going to take advantage of the conditions that are there. It's just a matter of, if they're nesting in shallower systems that typically aren't wet, but then, when they draw the waters down, those nests are going to fail. And we don't have the fate known, and that's what happened this year. Is we have 8 to 12 nests that we couldn't get fates to because the airboat trails dried out. We tried. We went with FWC with their boats, and we tried to get back into some of those areas, and it was just too dry. You needed an ATV or a swamp buggy to get back there. So in previous years, we've had them nest in 13, 14 feet of water, and we've had success with that. So it's just a matter of where the snails are, and the snail kites just taking advantage of what the water levels are doing. I might be able to produce a graph that just shows what Miguel did just over time, over different years, to maybe help clarify some of that, I guess, intuition for people thinking that it needs to be that high.
Yeah, and you kind of touched on this, Brian, but it's kind of a dynamic. There's short-term dynamics and long-term dynamics, and in the short term, it's good to have water levels high and you get good kite nesting. But we know what that does to the marsh, and eventually your productivity, your effort will go down if your marsh is degraded. And so we need those lows to rehabilitate the marsh and the conditions and rejuvenate the snails, and a lot of all those processes that are important in dynamic wetlands. And so, yeah, you can maintain the lake at 15, 16 feet year in and year out, and you'll get pretty good nesting for a year, 2 years, couple years. But it'll eventually fade out if you don't get the low water levels to reset things. And, snail kites are long-lived birds. They can live over 20 years. So, if a bird really likes nesting in Okeechobee, but we get a year or two where Okeechobee is not available to them, it's okay. We just, it might be better for the population to have a lower events every couple of years in Okeechobee to rejuvenate those higher year nesting efforts.
Paul
Yeah,
has
just-
had his hand up.
Oh, I just want to just quickly follow up with Tyler. Yeah, the big thing is, the change in water levels during the nesting season when nests have already been established. That's the big critical stuff. So, if we're thinking about drawing down water, then for a recovery effort, it might be best to do it before the snail kites start nesting. So there is a lot more flexibility with water level and water management.
Hey, Paul, did you have a different question, or do you want to contribute
No,
to
I
this?
was actually on the same topic. That
Okay.
slide, is it easy to put that slide back up real quick?
Yeah, no problem.
I was just kind of curious about, recession rates on the lake is a big deal, and we can't always control it because of irrigation. But the nest numbers are decreasing as you get into April and May, and I was just curious if you can tease out how many of the nests just got finished because it's later in the season, between how many nests might have failed because of rapid recession rates or getting stranded. Do you guys have a feel for that relationship?
I mean, that's something that we can calculate. I don't know from the top of my head, but we have the data for it. Yeah.
Yeah, because right now, with the lake, we've lost most of the submerged aquatic plants, and we think to get them back, we have to get down to the 12 or 11-foot range, which is not the best for kite nesting. And there's talk even this year of trying to do a recession, but we're at 16 feet right now. And if we tried to get all the way down to 12 feet from here, it would go screaming down, and that would probably be really bad for kites. And the trade-offs there are pretty bad. And quite frankly, I think we're probably too deep to try a recession this year, but some people have talked about it. And that's just kind of the question of how important is that recession rate to kite nest success as one of the things we look at. So anyway, just a thought. Thank you.
Thank you, Paul.
I saw that Brian Garrett had his hand up for a minute, and I wonder if if we want to know what's going to happen to kite nesting on Okeechobee, we can look to the SDAs that primarily maintain their water levels all year round. They don't really dry out, and we get when those are freshly inundated, we get kite nesting in there in pretty good numbers for 2 years, maybe 3 years, and then because they maintain wet conditions all the time, that nesting they go elsewhere basically. So that's a good example of what happens with stabilized water levels.
Yeah, just to add to that, we've actually seen nesting in the STAs in some years all the way from January to December. So, matter of fact, I think UF actually just at some point in December decided that they just couldn't keep going. So they actually took a break. But when they came back in January in that same cell, and this was back at I'm gonna guess it's like 2015 or 14. They came back in January and they already had chicks that were obviously hatched in December. So they nesting just went through the entire straight year. So I think we at one point had 18 straight months of nesting in a fairly small area. And it's just because you get a positive feedback. Although our operations managers don't always think it's so positive, but we basically would, once you have the birds in place, then we have regulations to try to keep the birds successful, and it just sort of like the birds keep coming until other things such as a tropical event happens, or maybe the foraging just stops being so good at some point. It's that point that the birds finally give up and move on to another site, it seems.
Yeah, and just to kind of add a note to what Brian was saying, it's like those STAs are super dense of exotic snails. So they have the food availability to reproduce year round, where with some of the other natural areas, you just don't see those densities as high. So they have that more natural decline in nesting over the time period. But where we have seen nesting go throughout most of the year are those STAs or more managed wetlands.
Go ahead, Holly.
This is Holly Andretta with the Water Management District. And, you know, a lot of times when we see that the snail density, where the snail kites are responding to the snails and the foraging, and we get that dense nesting, a lot of times it is when we've just after inundating like a new area or doing some kind of refurbishment, we've dried it out or lowered stages and rehydrated, and we don't know if it's a response to the flux in the soil that the snails are responding to or not. But it seems to be some kind of relationship where we get this boom, you know, population of nesters following the snail boom. And it's kind of a pattern we've sort of been seeing, you know, and I don't think we have a good understanding at all, but it's just kind of a pattern that might be a good study for somebody.
Are you saying that the district is going to let us sample snails year round in the STAs?
I don't know. We'd have to see a proposal and take it up to management. You know, we have the landowner agreement with limited take with the Southern STAs and 2 FEBs, and then we're working on a larger HCP programmatic for the whole footprint. Maybe that could somehow be incorporated into these, or with Zach Welch's umbrella contract or something like that, or just a study for a master's degree, some kind of thesis. But yeah, there seems to be some kind of relationship with bringing on new cells, inundating them and getting this response from the snails. And then we see the snail kite show up for a year or so until the snail population kind of goes back down to a normal population that we see everywhere else. It's like they're just having so much success with foraging that they nest there, and it's a second. They don't even have to search very far for a snail to get one. But then we have other areas like Nubbin Slough that's been online for a while, and we got nesting there this year. A little bit of nesting, but yeah, they seem to be following the snails for sure in our circumstance.
Yeah, and thanks for bringing that up, Holly, because I think that's one of the key things here, that relationship between nesting and water levels. It's one pattern, right? But nature, it's complex, and the biology of the snail kites, it's pretty complex. So there's a lot of different variables and interactions among different things that are driving this. So it's important to understand all of those, right? There's a relationship, there's a correlation between the two, but it's also important to understand those other intrinsicies. I noticed that we're a little bit on time for Dr. Ken Mayer to talk. I'm not sure if we should move on. I mean, I'm planning to be here for the whole session, so we can just continue chatting. I just want to make sure I'm not going over time for Dr. Mayer.
Now, with Dr. Wild not able to give her presentation, I'm not really worried about time in this first session. But thank you.
Okay.
So any other questions for Dr. Acevedo or general snail kite questions? Okay, hearing none. Dr. Ken Meyer, I saw you had your hand raised for a second. Do you want to move on to your talk, or do you have thoughts
no,
on this?
thanks. Yeah, it was covered. Thank you. But I'm not in any hurry, so would welcome further discussion if people would like.
I didn't hear any further comments, so I guess we can move on to your talk.
Okay.
Thank you, Dr. Acevedo, for giving that update.
Yeah, my pleasure.
Looks good.
Okay. You see it and you
Yep.
hear me? Okay,
Both.
very good. Well, thanks, everyone. This has been great so far. And Miguel, very nice to meet you in this limited but appropriate way, I guess.
Nice meeting you
Very
too.
nice job. And thanks, Tyler. Nice job bringing this all together and making it happen. I'm going to summarize my organization's work with monitoring apple snails for the Army Corps in Water Conservation Area 3A in 23 and so far in 2024. And then I'll end with a little bit on the snail kite tracking work that we've done over the years. We keep a small number of adults tagged, and that's been very worthwhile because as other things have changed in the kites environment, things with the kites movements seem to be changing as well. So starting with the snails, this is a contract for the Army Corps, with the Army Corps, evaluating apple snail population responses to changes associated with Everglades restoration, hydrologic changes. So I'd like to acknowledge my co-authors, Gina Kent from ARCI, my organization, Phil Darby from the University of West Florida, and Tiffany Trent, who does part-time work with ARCI. ARCI stands for Avian Research and Conservation Institute. We started in 1997, and we're still going at it as a nonprofit. Our mission is to provide data on birds that are imperiled or of great concern for one reason or another. Small populations, declining populations, changing elements in the environment, things that are critical to them, that sort of thing. And to push our data out in support of management actions. That's really our primary purpose. And these species, the snails and the snail kites, are a good fit, although we do very, very little listed species work, only snail kites and crested caracaras, as a matter of fact. A little pickup work here and there on eagles and peregrines and that sort of thing. So I'll give you a summary, first of all, here of the 2023 results for the snail monitoring in 3A. The traps are a meter square open box, top and bottom. And a trap throw is, you can't throw it very far, but you can drop it in the water and snuggle it down into the substrate a bit from an airboat. And we have 32 sites selected randomly with GIS in Conservation Area 3A. And around those 32 sites each year, we do at least 525 or so trap throws. At least 15 around each of the survey points scattered throughout the impounded area. So 15 throws. If we do not catch a snail, we continue through 15, and then there's a stopping rule that you can save time, invest it better if you stop sampling at 15 attempts if you haven't caught anything. If you have caught at least one snail in those first 15 attempts, you continue to 25 trap throws. So one of the big issues, of course, is what drives these population changes in apple snails. I should begin by saying there have been virtually no snail kite nests in Conservation Area 3A for at least a couple of years. And prior to the turn of the century, late 1990s, and we know this from work by the late Rob Bennett and others. Prior to that time, Conservation Area 3A had this glorious period of a few years where it was the center of abundance of nesting snail kites in Florida. And the birds that were produced there contributed to the start of the recovery, I guess, in the long run. Recruitment certainly went up with all of that nesting attention. However, it didn't last. And the suspicion was that water management changes made it impossible or less likely for snail kites to nest there in such large numbers, not impossible. And that what was driving that may be the snail populations, which of course, if you study snails and snail kites, you like to realize that it is a snail kite because it eats mostly snails. And so there's been a lot of attention in the last few years being redirected or directed by people who necessarily weren't doing work with snail kites directly, but a lot of attention, more attention directed to studying snails. So one of the biggest questions is the kites stop nesting in Conservation Area 3A or reduce their numbers greatly. And the Army Corps has been paying for monitoring of snails, apple snails, since that period. This work was started soon after that period. This work was started by Dr. Phil Darby at the University of West Florida, and he has become the apple snail expert. He backed off on the monitoring and the day-to-day work with apple snails, and we picked that up, picked up a contract to continue that for the Army Corps. So what you see, this is the bottom line of the results for 2023. Out of 565 trap throws, we captured 0 native snails and 9 non-natives. Now, the purpose of this study is to focus on native snails. Coincidentally, we get data on non-natives, and so we include it in these results. But the mission of this project is to track and understand the changes in the native snail populations. So once we adjust the density estimates using capture probabilities that are determined from trap to trap, so that comes down to a density for natives and non-natives combined, in this case, of 0.018 snails per square meter. So right at the bottom of this slide, what you see is a magic number. This is from the prescription for what it takes in terms of snail densities to support snail kite nesting. These numbers are not something that we started generating in 2017 when we started, ARCI started doing the snail work. These numbers have come from people who study snail kites and snails in combination, and they go back some time. So the target, the bottom end of the snail density range that will support nesting snail kites is 0.140. So as you can see, in 2023, we were way below that density. And in fact, there were no, to the best of our knowledge from US results, population monitoring and searching for nests so they can band young, no snail kite nests in 2023 or 2024 in 3A. They have been concentrating in the southwestern corner, and that was not the case the last 2 years. So I'm going to give you a look at 2024 data. We haven't done the annual report yet, but as you can see here, the situation is a little better, but not much. Still below the threshold at 0.033 snails per square meter in 2024 with 525 trap throws. I'm going to show you here what the adjusted snail densities, the magic number, since 2017 through 2024. And as you can see there, this is from our comprehensive 5-year report through 2021, plus our annual reports for 22, 23, and 24. Those numbers are consistently low. And in fact, there were only 2 years, 2018 and 2021, when we hit the bottom of the target, the prescription target for density of snails in 3A. So there's a lot of wonder and questions, and it's been confusing trying to sort out all the things that impinge on snail kite productivity and therefore snail productivity, and how that relates to water management, how the snail production relates to water management. So what we wanted to do is to try to contribute to that by looking at, first of all, how water managers can judge what the water conditions are in Southwestern Conservation Area 3A. So this magic range for snail density starts at 0.140 per square meter. But the range of depths, and this goes back quite a ways, the range of preferred depths for apple snail productivity and availability to snail kites, availability as prey to foraging snail kites. That range of depths is, people have come to agree on the 20 to 40 centimeter range. At 10 centimeters of depth, water depth, over the ground in a particular spot, the snails actually start to estivate. So we like to think that 20 to 40 centimeter range will make the snails productive enough to become useful prey to snail kites. And that 20 to 40 centimeter range also is a good depth for foraging snail kites, because in wet prairie, the snails come up to breathe on the stems of emergent vegetation, particularly plants that have the physical structure of Elaeocharis, for instance, a single emergent stem, fairly low density stands. So the snail kites can see the snails as they come up and stick their little periscopic breathing tube up just above the surface of the water. The snails don't come out of the water climbing up on the plant, on the stem. They just come up close enough to breathe, and then their suction releases and they fall back down to the bottom, because that's a danger zone. When they are up there just below the surface and the tip of that periscope is above the surface to take in some air, that's when the kites are snagging them. It's all a beautiful process and relationship and amazing to watch. Deceptively simple, but there has to be enough snails, and the vegetation and the water depths have to be just so to make that happen. So what you're seeing here, the blue lines here, the blue line represents the depths in 3A during this time period, as estimated from 3 to 4 staff gauges scattered around all of conservation area 3A. The orange line is what we actually would expect there to be in Southwestern 3A if we use those gauges, because those gauges that are spread out, and they're I'll show you where they are, those names, the names of those gauges in particular. They're off by about 20 centimeters in predicting how much water there is in the southwest part of 3A, where the kites were always nesting in the greatest abundance. So if managers want to track how water depths are doing for snails, we think they need to be looking at 3 gauges in particular. So I'm going to show you a map of 3A here and show you where these gauges are located. The blue triangles represent the staff gauges in the middle zone. These are 3 depth zones going from green to tan, in the lower right. So the staff gauges in the middle, and the red are the staff gauges that we think it would be better to monitor. The ones in red, the triangles down here, and one up here. This is all the southern district of 3A. So what we did was we created a circle here of containing 2 of these 3 staff gauges, and we wanted to compare depths at those gauges with actual depths that we were measuring in the snail kite traps when we sampled around 9 sampling focal points within this circle. This one is slightly out. There are 9 sampling stations for snail kites in our protocol. So what we did was go back and look at the depths from the throws, the trap throws around each sampling point. So right here, for instance, the crew would go there and start throwing traps within 200 meters or so of that centroid. This is just a reference point. And they would start throwing traps. And when they reach 15 with no snails, they would stop, or they would continue to 25 if they'd caught at least one snail. In the analysis I'm going to show you, we had 15 trap throws at each of these focal sampling sites because no snails were captured. So the 15 trap throws occur within a couple of 100 meters or so around the sampling point where the vegetation, the emergent vegetation in the water depths looks suitable for snail production and snail kite foraging. So what we found, this gauge shows you that same difference from this slide back here between the gauges that are spread out around 3A versus the gauges that are closest to that southwestern area of 3A. So here are the identifications of the gauges. And what we are suggesting people focus on is the Eden 12, Site 65, and W2 gauges in this map, the red triangles. So yes, what happens now, you can see the difference here in southwestern 3A. If we look just at the 3 gauges nearest that we recommend using, you can see the difference in water depth. So we could be out of the depth prescription there if we refer only on to those generalized gauges spread out. But we've also added to this graphic, these dots represent the average water depths for the 15 throws at each of the 9 sampling sites in 3A that I told you we were going to focus on. So these 9, they're actually dark purple, but these small sample dots. And what we found was that it's not a bad fit, that if you use these 3 gauges that we're recommending, and you're looking at this, it fits pretty well with what we actually would see in the traps when we're trying to estimate snail density. On average, it's a tad higher. So maybe that's a little bit of a further fine tuning, a correction of where we want to be sampling those 3 gauges, but with an understanding that it could be even deeper in the area where the snails are becoming prey to the snail kites. So from here, what we're going to do is go back more years and see how this relationship holds up. Are you using these 3 gauges in the Southwest? Is this giving us a good representation of the conditions for snails in terms of productivity and for snail kites in terms of catching them? So we'll go back in time and look at that. But eventually what we want to do is look at how well the kites are doing or the snails are actually doing in density. If these water conditions are being maintained in that important, very important February or March through June period. And we hope that will be informative enough. We know the water managers are eager to get this right, but there are complications to trying to manage water levels in such large areas of what is essentially a very, very altered hydrologic landscape. We have redesigned it, and now we're trying to recreate historic conditions for the plants and the animals that live there. And that's a tough one. I'd like to go back and look at what Rob Bennett wrote from his work that started back in 1986, his work on snail kites, and in 3A. So there is a monograph published by the co-op unit, Nesting Ecology of Snail Kites in Water Conservation Area 3A, that Rob did with Mike Hollify and Steve Beisinger. So I hate to put quotes up on a screen, but these are really important ones. This is Rob's experience based on his abundant experience. And I miss him greatly, just if for nothing else, because of all he contributed to snail kite ecology and understanding snails. What Rob determined was that he was finding snail kites to capture and to track 395 nests that he worked at. As long as these areas where he was searching have dried out once every 1.9 to 3.8 years. If they dry out more or less frequently, he learned that snail kites were no longer prevalent in those areas, at least not extensive nesting going on in those areas of 3A. So what this indicates is this area needs to experience some perturbation, disturbance, natural change, and hydrology from rainfall. But for this 2 to 4 year period, these sites have to dry down to be productive for snails and thus for snail kites. He also learned that the environmental correlates of nesting success were not consistent predictors of whether we were going to have these conditions. So, you know, that's an important thing to realize when we're chasing this magic formula for getting the water right for snails and for kites. It's a tough one, but that is the way it is. And what this suggests to me is that we need to accept some disturbance. We need to accept periodic being out of service. You know, I think everybody here realizes that if you watch areas that you get excited over places like Paynes Prairie and periods on Lake Okeechobee, Lake Toho, where everything is great. And the snail kites are being very productive because they have plenty of food. And then it stops for a few years. Fortunately, the kites are nomadic and they find other good places. It's, to me, managing snail kites is all about the network. It's about everything going on over the better part of the Florida Peninsula, at least the southern 2/3. That's what's going to determine whether we have sustainable populations of snail kites. Another couple of important quotes, I think, another one from Rob. The idea of critical habitat. I'll show you a picture in a second of where that is. But what Rob realized even back in the 1980s was that what was designated as critical habitat by the service in the 70s, and it has never been updated. We still have the same designations, and they're basically meaningless in terms of management. The protection of only the currently designated critical habitat would be insufficient to maintain viable populations of snail kites. Someone I've worked with in the past, when wood storks, Billy Brooks from the Fish and Wildlife Service in the Jacksonville office, he has a great way of describing how for wood storks, critical habitat is basically where the storks are this year. And so the whole designation and application of critical habitat for these water species. I mean, snail kites ecologically are very similar to wading birds in a lot of ways. So for these species, we need to be more flexible in where we site critical habitat and apply greater protection for the birds. And then this last quote, this comes from Buzz Holling and Gary Meffey, conservation biologists at the start of the discipline. And I think it's a very, very important thing to consider because it talks all about the importance of accepting variability and disturbance because they will increase resiliency. There were a lot of people who alluded to these facts when we began trying to restore the Everglades and things such as snail kites. And the fact that trying to command and control this system and do the same things year in and year out to make the system productive in all of its native species is just probably not going to work. So when we think about all the critical species in the Everglades and the species covered by the multi-species transition strategy, for instance, snail kites, wood storks, Cape Sable seaside sparrows, and also plant communities like tree islands and sloughs developing from wet prairie if they're inundated too much. When we think about those critical targets it would help if we could accept the fact that not all the best places are good in any given year. And that means that some years for some of those species are going to be bad years. But the only way we can capture a future for all of them is if we allow that to happen within the narrow limits that things used to occur naturally. If we have more of that vision. I think at the GEAR meeting, Mark Cook from Water Management mentioned that he was organizing a session about this very topic. And I'm looking forward to that. So I'm just about done here, but I want to run through real quickly. I got to talk a little about snail kites. I want to show you, this is just a collage of what we've been doing since some of the things we've been doing. That's Gina Kent in the top center. Out trying to catch snail kites to put tracking devices on adults. It started in 2007. We have an interest in mercury. We have an interest in long-term changes in movement patterns. So I'm going to show you here, we've got 5 adult snail kites with active transmitters, and these are 3 more that recently, 1 radio failed and 2 birds probably were hit by cars, actually, based on the evidence. Most of these birds are over 3 years in terms of time tracked, except for the most recent, the recent ones at the bottom. But we're in the 5th year for one of these birds, and another bird is almost there. So kinds of things that we're looking at, these are the 5 birds that are still active. You can see quite a difference there in the size of home ranges. Each color represents 1 bird, but these colors over on the right side, on the eastern side here, these are the most recently tagged birds. They just haven't had a chance to move around this much. But what the main thing I want to point out here, red and green are birds that were tagged on Payne's Prairie a few years ago, actually 4.5 years ago. And you can see they've had very long movements all the way down to the center of to the area on Lake Okeechobee that's been so important to snail kites. And they go back and forth. But over time, they've gradually been spending more time in central and south central Florida. There are no fixes below Lake Okeechobee. I want you to look at this map, if you would, please. This contains all of our GPS tracking data, first with satellite conveyance and then GPS delivery to us. So these are all highly accurate GPS fixes. That map on the left is based on data from 2007 to 2022. But look at all those fixes south of Lake Okeechobee. Those are old. Those are from the period up to about 2015 or so. And since then, and tracking work of juveniles by Carolyn Poley and Rob Fletcher shows similar. They're not going down there very much. And the juveniles actually in their first year make long exploratory movements. It was great to learn that from that work. But here you see all these red dots. That's what it used to look like not very long ago. And now none of the birds that we are tracking and for the last 3 years have been going south of Lake Okeechobee. There must still be a little bit of institutional memory in this population for the historic Everglades, for what was formerly called the Everglades snail kite, but that's probably mostly gone. What the kites have in their favor, though, is they're extremely good at getting up 1,000 feet or 2 and going long distances when the wind is good. And we don't see them doing this. We know it from the radio tracking of the remote tracking. They're very good at exploring places where they have been before to see if they're happening. Are there snails there? Are the water conditions good? And we have birds that have gone 150 miles or more and back in 2 to 3 days. They're checking it out. They're keeping track. And they're so good at that. And again, it's about managing this whole system. That's the future for sustaining snail kites and thus for understanding what we need to do for snails. Here's what the critical habitat map looks like. It was established long ago. There's except for Lake Okeechobee, there are not a lot of snail kites in these areas anymore. So I think it's time to start talking about this. I think it's past time and talking with the service. And as I wrap this up, and I want to thank everybody involved in this and this group, which has been so helpful in developing these ideas, talking with people at the Army Corps about the snail issues, especially, and how to get the water management right. It's been tough, it's been frustrating, but it's also been very gratifying. So, well, I'll just leave it at that and open it up for questions. But I think, oh, I want to say one more thing about one of Rob's conclusions, Rob Bennett's conclusions. What he mentions, he doesn't mention so much the 20 to 40 centimeter depth range for snails, but what he says snail kites need is 20 to 80 centimeters. And whether he realized it or not, I think what he was taking into account was what snail kites need is snails, so 20 to 40 depth, and what the kites need is deeper water for protection from predators. So I think that's important. We're looking for how to identify places where snail kites can nest. Well, not just the immediate nest structure, but when you go out to cover more of the foraging area around that nest, that 20 to 80 centimeter range is important, but it has to be variable within that range because the snails don't need or want it to be 80 centimeters. However, when the water drops below 80 centimeters, I think Rob Fletcher's team, and Miguel, you may know, I think the preference for water depth near nest is greater, more up toward 100 centimeters. Brian, you would know too. So anyway, that's an important thing to consider. Where do we have this range of conditions within the range that a pair of nesting snail kites want to forage? That's it. Yeah, I will leave it at that. Thank you very much.
Thanks, Ken. Yeah, I think it's a good idea to relook at the critical habitat. In the current climate, it's kind of amazing that the Kissimmee chain of lakes isn't considered critical habitat. It's the most consistent nesting area we have currently. If they stopped nesting in the Kissimmee chain right now, we would have years where we had almost no nesting in a year. The nesting there doesn't fluctuate up and down. We don't have really big boom years like in Okeechobee, but we also don't have years where we have almost no nesting. It's very consistent. So it seems to me like that's pretty critical habitat right now to me. Does anybody else have any questions for Dr. Meyer? Go ahead, Mark.
Hey, Ken, how you doing? Can you hear me okay? I can't hear you.
Ken, you're muted, but I can hear you, Mark.
So yes, I'm organizing this session at GEAR. Using it's kind of a trade-off session. I know a lot of people don't like that term, but we are looking at the ecology in a hugely changed system, as Ken mentioned. We've lost all that topography. We've got different water depths. We have a real issue, I think, with trying to restore the Everglades, for everything. We're never going to get back optimal conditions for all species that used to inhabit the Everglades. So, for instance, Ken's suggesting, in those good science in the 90s showing that we had this critical habitat for snail kites. And folks are probably not going to like me for saying this, but that was a disturbed system too. It was critical habitat at that time, but the 90s were not what the Everglades used to be. 3A was from what we can understand, was a much deeper, it had a lot more topography, deeper system, much longer hydro periods, and it probably rarely dried out more than once every 10 years. I mean, that we don't know for certain, but there is evidence to suggest that. So 3A may not have been great critical habitat. Maybe some of the edge regions and probably Everglades National Park and some of the shallow areas, but that area of 3A may not have been critical habitat. So anyway, I'm not saying it shouldn't be in the future. I'm just saying that things have changed and they continue to change, and we're going to see a lot more changes as the system, as we start doing restoration. So I just so if we dried it out, for instance, every, what were you saying, Ken? Every
2 to 4 years.
2 to 4 years. That would not be conducive to a lot of other critters. So that would not be very good for fish production, particularly larger fish for wood storks, etc. So anyway, so there are these sort of conflicting, when we look at these optimal conditions that we think these animals need. On the other hand, I do have a lot of optimism. I still think, we're still learning what wading birds need. We're doing this research now in conditions that these birds haven't experienced for 80 years, and from a wading bird perspective, and we're seeing that their needs are different than they were in the 80s, 90s, and early 2000s. They can use different areas, different depths. And also, I think we're still learning a lot about these apple snails and snail kites. We're getting new information all the time, particularly on the apple snails. And so I'm just wondering whether. Sorry, this is a long way round to talk about. We've got some, having a sort of a balanced ecology for as many species as we, to do well as possible is going to be tricky. But maybe we don't need to dry it out. Maybe we can have these, relatively shallow conditions, but longer hydro periods in the current system that could actually benefit snails, snail kites, and the more aquatic, longer hydro period needing creatures like alligators and wading birds and things. So what is the evidence that we think that we really need that dry down every 2 to 4 years? I guess that was my question ultimately.
That's based entirely on what Bennett saw and Justin working in 3A. And I think what he was referring to was local areas within 3A that would vary that much. Some of them, we're talking about a very small portion of the total area that, for whatever reasons, it does have an irregular bottom and water does pool up toward the southwest corner. He was talking about on a much smaller scale. And I know that I'm not a hydrologist. I can't imagine how difficult it is to manage for dry downs on a small part of a large area. But that is what Rob Bennett was seeing. And he's emphatic about that 2 to 4 year. If we get outside the bounds of that, the kites are not there nesting, presumably because the snails are not abundant enough. So that was the source of that.
But is that a habitat thing, do you think? Is it because it's creating this the more of a wet prairie type habitat rather than actually benefiting the snails, directly?
It may be. Yeah, it's hard to tease these different things out, but I think the point of the quotes that I included is that if we're trying to maintain the same conditions everywhere in an environment where that didn't happen naturally, not considering all the problems of scale when you try to look at this, that ultimately we're not going to succeed for some of these species. So.
Oh, yeah,
I,
agreed. You definitely
I
need
think about,
variability. Yes.
I think about wood storks. My group did the survey, aerial surveys for years for Billy Brooks. If 1 out of 3 years was bad for wood storks, I mean, like it could have been a total bust. The population was still growing. So I know that there are different species have different proclivities in this on this scale here, but just a way to broaden our thinking a little bit. When it comes to critical habitat and 3A being designated, I think what probably happened in the 70s was that people looked at Everglades National Park and they looked at 3A and said, hey, looks like marsh, looks like probably good snail kite habitat, and it's big and it's publicly owned. And if we designate it as critical habitat, it'll look impressive and we don't have to worry about development conflicts. Maybe that's what they were thinking.
Yeah.
Right. So I don't think it
Yeah.
was because they saw kites
One
doing
final
real
thing,
well there
just
in the
to bear in.
70s.
Sorry, Ken. So
No, go
sorry,
ahead.
I'll go off. One just other thing to bear in mind is that as restoration occurs and we get more flow, then we may actually see sort of improvements. You know, that the northern area of 3A is likely to improve for snail kites as well, because
Right.
the hydro periods will improve and yeah,
Yeah,
conditions.
and
will improve.
there are other factors. And I've been talking with Gina Roush from the Corps a lot, and I know she's talking with Tylen Dean, and these things are top of mind for them. Yeah, well, maybe not a good time to start a longer subject, but yeah, it is a real challenge for them, and that's why they're looking for better ways to track how these places are doing, places that have had kites in the past. There also are potentially other causes, as you know well, for the snail's decline, and it is kind of frightening. I mean, I know you have sampled in places where there are no snails, in Lila, for instance. So what about periphyton communities? Are they diminishing? That's critical to native snails. For those of you who don't know, it doesn't matter very much to non-natives. Eutrophic systems, deeper water, very little periphyton, that's good for non-native snails. That works for them. It does not work for native snails. So there are things like that, or calcium levels in the water, things like that. Cichlids, predation on snail larvae. So there are other things to consider, but water certainly seems like the place to start and see how much we can do. I don't. This is really outside of most of what I've spent most of my career thinking about. So it's fun, but don't rely on everything I say.
Can I stick a comment in here? It's Nick
Sure.
Allen, USGS. So I'm not a snail kite person, for sure, not a bird person, but one advantage of only very few advantages of getting older is getting to see the system change over many years. I'll never forget in the probably mid to late 90s when I was at the Water Management District helping someone tag egrets radio transmitters in the southern part of 3A. And getting there by canoe to where we were doing it. I'll never forget sitting in that canoe and turning 360 degrees and counting more than a dozen snail kites, just, flying around or sitting on dwarf cypress or other things. It just, when you talk about critical habit, I don't know what went into those decisions and where, but that memory is etched in my mind for better or worse. I've never seen anything like that since. Happened over a couple of days. Anyway, just totally anecdotal observation.
Oh, that's good, Nick. Thank you.
Danielle, go ahead.
Sorry, I didn't realize how dark I was. I'm poorly lit, but I raised my hand when we were talking about water levels and, what's the point of the dry down and the sweet spot for snails. And Mark, you mentioned whether it had to do with habitat. And that was always my understanding, is that it creates the type of habitat that snails need. They need the emergent vegetation to be able to breathe air. But when I was a student with Dr. Dorn, I put a bunch of baby snails in cages in 100 centimeter deep, 80 to 100 centimeter deep water. And they all lived and grew and did just fine. They were in a predator-free environment, but they were in deep water and they did just fine compared to snails that were in that, sweet spot range that Ken was talking about. So I guess just something to think about, that maybe predation does have a part to play as well in the water level recommendation. But yeah.
Well, I
Thanks, Cindy.
think you're absolutely right, Danielle. And that's what Nathan and Nate and me are showing at Lila right now, is that growth, this trade-off, this balance between growth and predation is essential. Unless these and predation, primarily actually from invertebrates, from other invertebrates, and so what we've shown and we're trying to publish right now is that unless these small, these baby snails can grow fast enough, they're just eaten up, and they they just cannot get to that adult stage. So there's this real fine balance between growth and predation, regardless of whether we have the right hydrologic conditions. So even under we've modeled it, so even under perfect hydrologic conditions, there's not a growth in the population unless you can have this fast growth rate to be able to outgrow the predation.
Right.
So, and that predation might be a function of the way we're managing the system. We don't know.
Yeah. Well, thanks, Mark. It's nice to hear about what's still going on out there.
Yeah. No, it's pretty cool actually. Is Nathan and Nate on today?
I think Nate Barris is on.
Yeah.
I didn't see Dr.
I'm
Dorn.
here. Yeah, I'm here. I'm just, I think you hit it right on the head, Mark. Good job.
Thanks, Nathan. Yeah, it's your little child. That child, this one is excellent. It's stunning. It's brilliant work that what Nathan's done. Actually, I really hope we get it published soon because it's really exciting stuff.
This is Ken. I just wanted to call attention to a question from Hillary Flower. Maybe Brian Jeffrey could address this. Benefits of critical habitat. So from the services point of view, anybody want to take a stab at that?
Hey, Ken, this is Tim. Can you hear me?
Okay.
Oh.
You go ahead, Tim.
Okay. Can you guys hear me?
Yeah, we got you.
Okay. Yeah, I saw this in the chat. Sorry, I've been hopping off and back on throughout the meeting. And so I missed the conversation on critical habitat, but I can just maybe give you a general overview. So designating critical habitat, does not really have an effect on activities that are conducted by private entities. It's really going to have more of an effect on federal agencies, on federal properties. But there are a number of benefits to designating critical habitat. I mean, just the act of identifying critical habitat sort of informs people, what areas are important to species, to their conservation and recovery. It raises the awareness of the needs of those species. So there's benefits regarding that, but in most cases, when from a endangered species consultation point, we're going to be consulting on the species wherever it occurs, and so the benefit of designating critical habitat is more, not necessarily from the regulatory standpoint, but more from kind of the awareness standpoint, I would say. Those are kind of my thoughts on it. if that was the question.
Yeah, so Tim, the question was more related to WCA 3A is no longer has any birds, but it's been designated critical habitat and it's still designated critical habitat. While the majority of the birds are now up in like the Kissimmee Basin, St. John's Basin, other locations, Paynes Prairie, which are not designated critical habitat. And so why are we not designating those areas as critical habitats? And why do we maintain the 3A as critical habitat? And, I'll just add to what Tim said. We in the Fish and Wildlife Service, we manage the species where it is. And so no matter whether the birds are in critical habitat or outside of critical habitat, there's still the regulatory guidance of the Endangered Species Act that allows us to address the conditions that the birds are utilizing. The designation of critical habitat is a regulatory process, and it requires, an entire process of evaluating the critical habitat and redesignating it. Right now, we're in the process of designating critical habitat for a handful of species, not snail kites, but bats and other species, because those are on our list of things that have to be done because of court guidance and requirements. So probably the reason why we haven't reevaluated critical habitat is because of the workload associated with it and our inability to move past what we have to do to what we want to do. So that's probably a large part of the reason why it hasn't changed in the past or in the recent history. The other thing is the regulatory handle on critical habitat is the Tim, what's it called? Adverse mod of critical habitat. So the project cannot result in adverse mod. However, what we've seen over time is that we've had a continual degradation of 3A. The habitat is, no longer utilized, and it's not suitable for snail kites. We just can't say what caused that. Was it a federal action that caused the habitat to degrade, or was it the climatic conditions and just the general operations and things of 3A that are causing that habitat to degrade? So we have no real regulatory hammer to go after an agency or something, because every time something happens, it's because of a hurricane or it's because of a significant rain event, or one in a 100 years rain event or something like that gets blamed for the high water issues. Does that help answer that part of the question?
Yeah, thank you, Miles.
Yeah, we would love to be able to keep up with where the species is and modify critical habitat. It's just, lack of staff, lack of resources, lack of time, and just not able to get it on the list of things to do. That'll save Brian from answering since he's only been with us for a month.
Thanks, guys. I appreciate it.
Do we have any more questions for either Ken or really any general snail or snail kite questions? We got about 10 minutes before lunch break, and then we'll reconvene at 1 o'clock. So, this conversation's been pretty good, and I'd like to keep it going until we have to break. All right, we've got a question in chat. Wondering if there's data available and shareable for locations of failed snail kite nests. For instance, in 2024, 9 of 11 nests failed in the St. John's Marshes. Is location data available for these nests and or failed nests in the past? We would like to be able to look into potential correlations in this area if the data is available, if no one else is currently doing so. Yeah, there definitely is.
I can take that, Tyler, if you want.
Yeah, go ahead.
Yeah, so Christine, you might want to talk with Diane Hall. She's the person who has a contract with the University of Florida that funds some of the monitoring and nest monitoring of snail kites. She should have all the historical data for a while. And if not, I'm sure you can contact Miguel and he should be able to provide you with some information that you need.
Yeah, just contact Diane and you can let us know if you need anything else. We'd be happy to help.
Perfect, thank you.
And if you're focusing on cattails, FWC has done a lot of cattail management projects that have had real positive results with snail kite nesting on them, especially particularly in Lake Okeechobee. The non-native snails really seem to respond to removal of cattail. But if you're working in big cypress marshes, then I think there are non-native snails there, but you also have a fair amount of native snails, so it might be different objectives. Paul's asking, he still says, I think a survey of parasites and native snails, maybe from exotics, would be useful toward understanding why natives are still doing so poorly in the WCAs. Ken, do you have any insight into parasites in native and non-native snails?
I do not. I would call Phil. I know that he's been interested in that. Phil Darby. Sorry, I can't help you out.
I've heard of this in some of the snakes, native snakes getting parasites from exotic snakes. So if we could check on that, that would be maybe tell us something. So thanks.
Yeah, if you guys aren't up to speed on this, there is a lungworm that they believe was brought in by non-native, I think, pythons, and can be carried by pythons that are affecting some of our native snakes as well. Does anybody else have any comments or questions?
I got a quick question for Ken. Ken, have you seen any patterns of where you have seen the natives versus non-natives within the WCAs? Are you seeing any that are closer to the canals or any that are more prevalent in the interior?
No, nothing that we've noted, and that's part of the problem of just not having many opportunities to, it's just amazing to throw 565 traps and come up with 9 snails, over a couple of 3 months. So yeah, I wish I could tell you. That's a good question.
Yeah, I keep wanting to, or keep intending to, and I've been having conversations with people about, maybe we've had good conditions in certain areas for a period of time, but the snail density just wasn't there to really establish, good population. And I'd like to get some modeling done to say, okay, under the new regime, hydrology regimes, these areas should maintain pretty good hydrology for native apple snails, and then look into cultivation and seeding of those areas and see if we can kind of jumpstart some areas with a good snail population, introducing enough snails out there they can find each other and reproduce in high enough numbers to support the population. So anybody has any thoughts on that, I'll welcome them.
I can add a little. Oh, sorry. So Danielle, go ahead.
I just was thinking, wasn't the Miccosukee tribe putting snails out somewhere once upon a time? Are they still doing it? Oops.
Hey, this is Marcel with the Miccosukee tribe. So yes, we still do have the apple snail rearing facility. We're we're releasing a couple thousand snails per year, primarily within the federal reservation, and we're doing so pretty selectively, right? We're trying to figure out areas that historically didn't have the right hydrologic conditions for the snails, but as water management operations change and restoration moves forward, it's now better conducive, and we kind of use the snails that we reared to establish the snails in those areas. We monitor their success, and we found that within the federal reservation where we're releasing the snails, they take quite well and they persist for as long as, you know, the years that we've been monitoring them. So we've had good success there, but yeah, it's definitely a smaller scale effort, right? We're doing so on the federal reservation, and we don't have the facility for a wider landscape scale contribution at the moment.
So, Mark, I see you have your hand up.
Yeah,
go ahead, and then
we're actually doing a little study in Lila on this right now, looking at sort of what sort of densities of snails are needed to actually kickstart a population, a viable population. So yeah, we're working at it right now. So hopefully we'll be able to provide some more information on this in the near future.
That's great. Marcel, I had a couple questions.
Yeah, I did present a little bit of those preliminary results in the spring, I believe. We did another round of introductions this year, and we're looking to see some more results to finalize those results before we talk about it more in depth. Yeah.
Nathan, are you going to talk about that at Gear?
Yeah, so yeah, that might be something about in Gear. Looking at some of the old historical records of apple snails from the Maud Waters data in the national park, and also a little bit into the WCAs as well, and some of our sites there. And then looking at perhaps if there may have been any extirpations, and then what that might mean for restoration and these kind of trade-offs.
Marcel, I had a quick question for you. When you say you're doing these introductions and then you're monitoring them, how are you monitoring the success of the reintroductions?
Sure. So yeah, we don't have an extensive staff to do the full monitoring approach we would like. Basically, what we're doing now is, we obviously mark where we do the reintroductions, and then we return to the sites periodically and just kind of do a density counts, right? Of walking around the marsh, surveying how many snails we see, and we compare that to prior surveys, including surveys from before we did the introduction, to see if we actually boosted the snail density locally, and if that is a sustaining trend.
So you're counting snails and not egg clusters?
Correct, but we egg clusters as well.
Okay. And either you or maybe Brian can answer this. UF does monitor kites across the southern 3A landscape. I'm not sure they get into the reservation or able to get in the reservation. So if there were kites nesting in the federal reservation, I'd like to know that, if it's possible that data could contribute to the overall understanding, whether it's allowing UF to get in there and do the monitoring or sharing of information. Do you think that's possible?
It's something I'm definitely willing to try to come up with a more defined plan and bring forward to the tribe and see if we can get that approved. I realize the importance of it, and UF definitely has the more expansive, ability to monitor this stuff than we do. We're just spread so thin with all of our other projects. But I'd be happy to get you guys in the tribal reservation and assist with that. But yeah, it's just something that would have to go through the appropriate kind of bureaucratic hoops and get approval that way. But follow up with me. Let's see if we can get that coordinated.
Yeah, and just to follow up with Tyler, the UF does respect the boundaries of the Miccosukee tribe, and while doing surveys, but I'm sure Miguel would be more than happy to collaborate with you guys to collect the data.
Yeah, I just wrote a message here in the chat that yeah, we'll be super happy to work with you. So yeah, I'll write to you and we can schedule a chat.
Great, thanks. With that, we're at our lunch break, and then we can reconvene this meeting for 2 more discussions at 1 o'clock. Okay. Thanks, everybody.